looked through the auxins and given you a sample of what they can do for the plant. And this is not by any means exhaustive. Auxins can do much more than what I have time to carry through on this class. But this idea that they're produced quite frequently in the shoot tip, excuse me, and then that is helping with apical dominance. So this idea that if auxins are in the shoot tip and they're flowing down the stem of the plant, they keep the axillary buds quiet until that signal gets released. And the way to release that is essentially to get rid of the shoot tip. And then that releases the axillary buds. They start to produce new branches or leaves. And you can get a difference in the overall shape of the plant under that mechanism if you want. We looked at the idea of adventitious roots that we see, like that powder that you might buy to do where they say, you know, root, I think it's called root tone. That's one popular one, where you take stem cuttings and then dip it in the cut stem into that, and it helps with growing roots. It's actually a lot of auxin in that powder that is doing that. And so that helps. And then also we use auxins in tissue culture techniques where we clone plants in special containers that have tissue culture medium, and the hormone in that medium then will promote what we call callus formation, which is a bunch of plant cells that just haven't differentiated yet into, you know, shoots and roots and leaves. And so that's kind of like phase one. You want to just get a lot of good growth, and then you spike in a different hormone, and that tends to take that callus tissue and get it into the different steps of roots and leaves and stems. So we looked at that. Phototropism, this idea of the plants growing towards unidirectional light, like the light coming in from a window, that bending toward the light. Phototropism, auxins are really in play there. And I'll talk a little bit more about that, and gravitropism as well. So I'm going to come back to phototropism and gravitropism in a bit. So let's continue on with the second group I wanted to mention, which are the gibberellins. And again, S on the end, because there's many different chemicals that fall into the gibberellins category, okay? And so some are synthetic that have been made by humans that are doing research and want a little bit more stable gibberellin-like molecules. So, and then there's plenty of other options that the plants are making. So this is a famous picture where you're looking at the cabbage plant, and you can see that the ones on the right are quite dramatically taller. So what happened was when they, you know, it looks like there's what, four or five plants that they started with, and what they did was they kept two plants, they didn't do anything to it, and then the other ones they sprayed the shoots and leaves with gibberellic acid. That's why it's called GA. Gibberellic acid is another word for gibberellins. And so you can clearly see stem growth is a huge thing that gibberellins do for the plant. And so as the plant is growing taller, gibberellins are part of that. And you can demonstrate this. We do this experiment in Plant Fizz all the time, and it works beautifully, where in Plant Fizz, what we do is we take pea plants. So there's a version of pea plants that are called dwarf pea plants. And that's what you're seeing on the right picture here, or the right pot, where you see these really short pea plants. And then what happens is you can spray. So both of the pots started this way, and then they sprayed the left dwarf plants with gibberellic acid, and you can see this big increase in stem length. And so the reason they're dwarf pea plants is that they don't produce their own gibberellic acid very much at all. They produce like 1/20th what they really should produce, but they can still respond to gibberellic acid. So if you spray them, they'll go, oh yeah, and they'll respond accordingly, which is to grow really tall. All right. And so stem length, gibberellic acids with intact plants like that, you can see stem length. Now, Non-dwarf plants, sometimes the response is modest. It depends on the plant, but in general, you can say safely that gibberellic acid does allow for stem length increase. Here you're looking at grapes, and the grape industry is huge globally and for many different types of grapes. And so here you're seeing two different bunches, and I think it's obvious that the ones on the left are smaller grapes. Generally, when you sell grapes, if they're smaller, they're a little bit less sweet, not as attractive to us when we go to buy them in the store. We tend to like plump, big grapes. And so basically what they can do while the plant is producing their grapes, you can spray the stem tissue where the grapes are forming, and it helps the stem tissue grow in length a little bit so that the grapes have just more physical room. And when you give them a little bit more room, then they plump up and they get sweeter, bigger and sweeter, which is more profitable. And we tend to like that as we buy it. So that's another big application of gibberellins. Here you're seeing this idea of a seed germinating, which you're doing in lab. And so this idea that gibberellic acid is part of the process to get seeds to germinate. And so that's going on right now in the seeds that you picked and germinated last week. Hopefully they're going through this process. So when you come in the lab tomorrow, you should definitely look at your plants, water your soil, make sure they're happy. Another thing that gibberellins can do for the plant is this shift in its life cycle from its juvenile phase to adult phase. So that's analogous to puberty in humans and then mature adult. Okay, and so this idea that plants have their own puberty where they, before you go through puberty, you can't produce viable offspring. And it's the same thing with plants. In the juvenile phase, they just can't do it yet. And so, of course, if you think about that's a huge step in the plant's life, going from immature to mature. And so the gibberellins playing a role in that is very important. Okay, so gibberellins are another big group of hormones that the plant is producing. And again, depending on which aspect you're thinking about, it's produced in different tissues. It could be seeds, it could be stem tissue. We do see it in actively growing regions of the plant body in general. Okay, cytokinins are another group. Cytokinins are very important in the process of going through cell division correctly. And so with this idea of mitosis, we're just looking at, cell division and growth of the cells through just replicating. And so the cytokinins are known to help with that. When we work in tissue culture practices and clone plants through tissue culture, cytokinins are the other huge hormone that's regularly involved in this tissue culture process. We do tend to see that it can be produced in fair amounts in the root tips, but it's also found in other regions of the baby plant, the embryo. So the embryo's in the seed. You're looking at a picture, by the way, of a root tip. And so when you look at some of the other things that the cytokinins can do, the lateral or axillary bud growth. So once those axillary buds have been released from the control of auxin, that apical dominance, then the cytokinins can kick in and also kind of stimulate the axillary buds or the lateral buds. They're also really important in chloroplast development, which, for a plant, having good functioning chloroplast is pretty important. And so to get that chloroplast from, its initial, it's just a plastid, and then it starts to morph into a chloroplast, getting that whole thing smoothly working all the way to a mature chloroplast, you have to have cytokinins. And again, they've tried to make cytokinin deficient plants like they have with auxin deficient plants. And with both of those hormones, they cannot get a plant to be cytokinin deficient. Not that I'm aware of, I keep checking. And so auxins and cytokinins are just so pivotally important to the plant that you can't even make a mutant plant. It quickly dies if it's deficient. Cytokinins are also known to delay senescence. Yes. Yeah. Yes. Not really. Yeah, there's kind of separate entities, but you're right. I do have that notes that. So it's kind of like with apical dominance idea, the auxins and cytokinins are like at odds with each other in a way, right? So auxins are promoting apical dominance, but the cytokinins can suppress it. And so basically what we tend to see is once the axillary buds start getting active and growing, you tend to see there's a big downshift in auxin and an upregulation in cytokinin. And then that keeps the axillary buds moving along and growing appropriately. So sometimes you will see that hormones will kind of be at odds, and which kind of makes sense. It's kind of like sometimes you need to press the gas, sometimes you need to press the brake idea, and so you'll see these hormones kind of acting in opposition sometimes. Senescence is a fancy word that just said programmed. It means programmed cell death. So all cells eventually will die, but when you think about a multicellular organism, like some of our cells are dying right now, or like our epidermal cells, they're dying and sloughing off. The lining of our intestinal tract, those cells replace themselves totally, like once a week or something like that. And so, that it's just normal. However, cytokinins can be there to kind of slow the process down under the right conditions. And so here I'm just showing an image of beautiful fall colors because under natural conditions with deciduous trees that drop their leaves in the fall, what you see happening is cytokinin levels drop. And then that causes the shift into them going, okay, I guess it's time to senesce. And before they senesce, they give us these beautiful colors before the leaves fully drop off. Okay. So if you have a tree, for example, in your backyard and you're like, I want it to not go through fall color change, theoretically, if you knew the right dose and applied it at the right time, you could delay your tree from turning its colors that it does if you want. You have to get the right dose. The picture on the right here is trying to show the same plant, one that is senescing and the other that isn't. And so I think you can see, especially down here, this plant is looking kind of sad and it's dying off. And so the one on the left, I believe, was engineered to overexpress cytokinin. And so it's just delaying the senescence process. So we do this experiment in Plant Viz where we take leaf discs, we cut little, even like a corkboard, we cut leaf discs and we soak them in different solutions, different concentrations of cytokinins. And we can see that with the right dose, you can get those leaves to stay green and healthy, even though they're floating around in the dark in this liquid solution. So that is another group of plants that are hormones. Did I put in there? So you should put in your notes. I mean, I did say it verbally, but make sure you have in your notes. I thought I had it in here, but I guess not. Maybe I put it in later. That cytokinins are involved in tissue culture. Make sure that you understand that. So auxins and cytokinins are the two big hormones in tissue culture work, where you go to clone a plant and grow it on that artificial medium. Okay, another hormone that I wanted to mention is abscisic acid, and that's just one chemical. There's only one abscisic acid out there. So this one we tend to think of this hormone as a brake instead of the gas pedal. It tends to have a lot of braking action. So it has inhibitory effect on many other hormones. And then under the right conditions, it can, well, let me give you some examples here. So in fruits, for example, as the seeds are developing in that fruit tissue. So once the flower gets pollinated and fertilized, and that, the egg in the flower has been fertilized with sperm, that then kicks into fruit and seed formation. For a plant, you don't want the seed germinating right away. You want to get it to maturity, but then don't continue on into further growing. You want the seed to kind of take a pause, so to speak. All right. So you want the fruit to develop, you want the seed to develop to the point where the baby embryo is in there and it's good to go, but you don't want the seed to germinate right away. With the idea that if you're the mother plant, you want your seeds to get spread out. You don't want all your fruit to drop and grow right by you, because that's a lot of competition. So you want that fruit to get gathered by other organisms and get taken away, hopefully get your seeds out there in different suitable habitats. And so when you look at a seed, the seeds that you worked with last week in lab, they had a fair amount of abscisic acid in them. It's just saying to the seed and embryo, be quiet. You're good to go, but just be quiet and don't germinate yet. Now, when you put them in the soil and you watered and got them all wet, then what's happening is that's a signal that, okay, we got a good environment, we got moisture, but it's also diluting out, so to speak, the abscisic acid. As the seed takes up that water, it's partly diluting effect. And so the abscisic acid levels go down, gibberellic acid goes up, and you start to get some nice germination. It also is keeping the buds quiet here in the wintertime. Mm-hmm. Yes, and so some seeds, especially gymnosperm, conifer trees, they won't. Their seeds aren't going to germinate unless they go through a fire. So heat, essentially, they have to have enough heat hitting the seed, and in that case, that's like an extra stimulus along with the hormones. I mean, we still see the shifting hormones, but first fire, then you start to see abscisic acid levels start to go down after that. Yeah. So those are special conditions for sure, very unique. So seed and bud dormancy, it doesn't make sense for these trees to have their leaves develop now. It would be detrimental. And so this can be really, really important, especially as we get closer to spring in this area of the country, where the trees have to be paying attention to actually a lot of environmental signals so that they will get their buds germinating appropriately. And sometimes they make a mistake. So for example, if the weather warms up and then all of a sudden we have a huge cold snap, the plant was kind of faked into it, like it's spring. And so they start to get into, their buds are growing and forming new leaves, forming some flowers, and then cold snap hits and then they're in trouble. And so that has happened, it can happen. And the tree, basically, that flush of leaves are now, usually they die off because they freeze. And then the plant has to try to recover and regroup in early to late spring. And that can cause problems. It can cause great delays in growth. And then their overall growth for that growing season tends to be delayed quite a bit. So having that signal, so abscisic acid is part of that, like just stay quiet, stay dormant. Abscisic acid is also very, very important to the plant in water stress conditions. And so if you think about it, if you're a plant and it's, the growing season, you're actively growing and your roots are in the soil, and then you start to, go through a drought period, it's your roots that are going to be experiencing the drought first, right? Because that's where they're taking up the water. They're getting water through their root system that's, coming from the soil. And so actually what we see happening is as the plant is sensing, oh boy, we have less and less water, we're getting into a drought situation. What happens is that the abscisic acid actually gets made in the root in response to drought. And then that abscisic acid has force all the way up to the leaves. And what are we looking at in these images? What are those? Stomata. Yep, very good. Stomata. And so they get all the way up to the leaves, get to the stomata and say, shut down. All right, close over the pore, because they're sensing that drought and they want to conserve water. So they'll shut down their stomata in response to that drought. But it's actually down in the roots that the sensing of drought takes place. The abscisic acid synthesis takes place down there. And it actually stimulates, so it shuts down the stomata in the leaves. But on the other hand, abscisic acid that's being produced in the roots is telling the roots, you should probably grow. Go search for more water. And that's what happens. And so you will see, if you look at video footage of plants undergoing drought stress, you will see that their shoot and leaves stop growing and the roots are starting to search, which makes sense. It's like, hold off on above ground growth because we're in a crisis here. We need more water. We need to go search for it. And so they'll grow out their roots to try to find more water. Okay, so it's kind of a dual action for this condition of water stress. Okay. Another hormone to think about is this one, ethylene. Ethylene is, so when they first started working and hunting for hormones, they came up with what they call the classic five, which that's what we've been covering. The auxins, the cytokinins, the gibberellins, abscisic acid, and now ethylene. So this is of the classic five. This is the only one that's gaseous. It's a gaseous hormone. And this one, oh boy, pretty much produced anywhere in the plant. It's hugely important. And it really, really helps the plant deal with stress. Different, whether it's pathogens attacking the plant, that kind of stress, so biotic stress, or abiotic. Abiotic means non-living things like drought or flood or cold. All right, so you have these different abiotic stress. It can kind of snowball. So once it gets going, it can start to feedback on itself on the pathway and produce more and more of the ethylene. And so it can trigger its own production. And this is something, it's a gas, but they know the pathway. And so what they can do in like real world applications, what they can do is there's a liquid chemical, a chemical that they can dissolve into liquid, and they can water the plant with that. And then when the plant takes up that water with the chemical dissolved in it, it quickly converts it to ethylene. So there are some handy ways to work with ethylene, even though it's a gas. And so, for example, in the commercial crop production industry, they will use ethylene to ripen up fruits. And so it's a way, and they can modify plants to like hold off on fully ripening until it gets shipped to its destination. So they've been working on that, like with tomatoes, for example, shipping ripe tomatoes. Very challenging because they get bruised so easily and break down. Sometimes they'll ship tomatoes green, and then once it gets to its destination, they'll expose it to ethylene and get it to ripen up. So the other thing it can do, this other kind of thing, is that it can cause leaf abscission. So every year, trees around here go through leaf abscission. Abscission means, in this case, it's the leaf blade that's, well, the petiole tube. It's going to fall off the tree. And so this is a natural thing for trees every year in the fall. After the beautiful fall color change, ideally you want those leaves to fall off and but don't have them fall off till you've taken all the nutrients out of them. So in the fall, plants are really busy. The trees are really busy. We get, this glorious visual fall color change to enjoy, but as we're enjoying that, those trees are going through a lot of activity because they have to take all the yummy stuff out of the leaves, all the useful stuff, and bring it down into their roots primarily, and sometimes the stem tissue for storage. And then after they've done all that, then it's usually to the point where the leaf goes brown, and then the abscission zone, there's like a break zone in the petiole of the leaf that will weaken and cause it to fall off the tree. So here you're just looking at an experiment where they took holly leaves and they put a beaker inverted over them, and one beaker is just normal, the one on the left, they didn't do anything to it. The one on the right, what they did was they put, they didn't show it, but there's a cut on that apple. And so with ethylene, you may want to put in your notes, I thought I had, ethylene really gets upregulated in response to wounding of the plant. When you wound the plant, or a wound can be something that you do on purpose or accidentally to your plant. Maybe a bunny rabbit comes along and starts chewing on the plant. That's going to release ethylene. So it could be a mechanical thing, that maybe you take a stem cutting. That's a wound. You created a wound. And so at the cut areas, you're going to get ethylene release. So when you wound an apple, and then put it in with the holly plant and then capture the gas, the ethylene that's getting released by inverting the beaker. So now you're flooding that holly plant with and that causes leaf abscission. And so ethylene is upregulated in response to wounding as well as all kinds of other stresses that a plant is experiencing. So if you want, you can take advantage of this. If you go to the store, sometimes all the bananas that are on option are green. It's frustrating. It's like, there's not a yellow one in the whole pile. So you can do this at home. Get the green ones, take an apple and cut it, and then put it into, I tend to use plastic because paper bag instead of plastic, just because it cuts down on fungi attacking. Because if you get too much condensation, sometimes you can cause problems. So paper helps capture the ethylene, and then you can get it usually by the next day or so. You can get it to green up. So there's an easy trick. So some fruits will respond like this, like bananas are famous for responding to ethylene that you provide to them. Other fruits though won't. So it just depends on the species, whether or not they're going to respond. So stress is a big one, whether it be, like I said, something in the environment, like, flooding, for example, the field got flooded, or if you take a stem cutting. So if you think about it, and then auxins and cytokinins can also upregulate ethylene. So as those two hormones get active, they tend to trigger ethylene because of the way ethylene is needed for these different stresses. And so this is another thing where I think it's really, again, I give a lot of credit to the plant physiologists who study hormones, because there's all these interactions between hormones that they're trying to tease apart and figure out. The image that you're seeing here are two containers of cut flowers. So the one on the left is sitting in a beaker of a solution that has this STS solution, which, let's see, not low enough. Maybe where you're sitting. That's low on the PowerPoint slide here, or the screen. But it says STS. Well, there you go. Right. Let's see if that does it. So the STS, that's thiosulfate, and that is a chemical that is known to essentially sop up ethylene and kind of keep it it binds to ethylene, so ethylene can't act correctly on the plant. And so the one on the right didn't get that treatment, and you can see the cut flowers are looking pretty sad. You wouldn't want to buy those for Valentine's Day, right? So this idea, if you own a flower shop, this is a big deal because you have, I mean, think about it. When you get your so many of the flowers delivered to the flower shop, they're already cut. And if you have a bunch of ethylene going off in your shop, you're going to cause the mess of the flowers. You're not going to make much of a profit, and you'll start to get a bad reputation as giving people flowers that are going to die really quickly. And so this is a big deal. There are several different ways you can try to minimize ethylene impact by certain chemicals. They've worked it out pretty well because this is a big industry. I have a friend who used to own a flower shop, and this is like, she was down on all that. She knew exactly, ways to do this correctly so that her cut flowers stayed nice and fresh for days and days and days after the people bought it and walked out of there. So what you're seeing here is flowers are essentially modified leaves. So you're seeing enhanced leaf senescence in the presence of ethylene. And so it speeds up, in this case, the flower death. And so again, if you're trying to make a profit off that, you kind of want to minimize the presence of ethylene in your crop. Okay. So plant hormones, if you take plant physiology, we go into more detail on each hormone. And then we also, at least when I teach it, I like to also show how it's being used commercially in real world applications. So we do expand on this in plant physiology. And I'm happy to chat with anyone that may have additional questions on this. But the next topic I want to get into with plant growth is, okay, so now we know that plants have hormones that are, really precisely controlling their different responses to the environment, their life cycle. So, but we also have to think about some other aspects of plants and how they operate, I guess, in their life. And so one of the big things to think about is plant movement. And this is the scan on the front of a book. And you can see Charles Darwin and his son Francis are the authors. And so they were one of the first, again, to really kind of do some nice research on how are plants moving and how do they do that if they don't have muscles like animals do. All right. And so hormones are definitely involved in this, but we need to think about some other things as well that come into play. And if you think about it, a plant needs to be able to move in order to respond appropriately to its environment and to create situations that are good for growth and reproduction. So it does make you kind of sit back and go, yeah, how do they move without muscles? We're so used to thinking about movement is muscle driven, and they can move. And sometimes our eye doesn't pick up on it that easily because sometimes their movements are quite slow, right? So you need to take video and then speed it up, and then you'll see, oh yeah, they are moving. It's just that, they move on a time scale that's much slower. And so if you think about it, there's really two major mechanisms. One is turgor movements. So this is the idea where plants manipulate their turgor pressure within their body and tissues. And when they manipulate it the right way, it can cause different parts of their body to move. And sometimes it can be very dramatic and quick. Like we can see it happen in real time very quickly. So when we say turgor movements, it's just water pressure that we're talking about. Okay, and we looked at vacuoles just the other day and how the big central vacuole in a mature plant cell is so important because it creates turgor pressure, water pressure. So this is kind of what we're thinking about. So often what we see is that a region of tissue will go through turgor pressure, but not the whole region. Like part of it really gets increase in turgor pressure, and another part gets a decrease in turgor pressure, and that will cause the movement. And so that's being shown on the right here, where this is a petiole of a leaf. And what they're showing here is that the left side here where my laser, can you see my laser pointer? Okay. Okay, that side is not getting much turgor pressure, but the other side plumped up with a bunch of water and turgor pressure. And so you have the right side really plumping up with pressure, the left side not so much, and it causes that little bit of bending like that. Okay, and so you can see that. And then with the image on the bottom, those are guard cells guarding the pore, stomata. And what you tend to see here is that it's turgor pressure that's causing the guard cells to either be closed, if there's very little turgor pressure, the guard cells are closed over the pore. When water rushes into the guard cells, it actually causes the guard cells to, because of the turgor pressure and the way that their cell walls are designed, it actually causes the guard cells to kind of move away or pucker away from each other, and then that reveals the pore. And so again, it's a turgor pressure idea. And so it's kind of an easy mechanism, pretty simple mechanism for the plant to I want my pores open. So I'm going to rush water in. Well, maybe 5 minutes later, well, now I need to, change my mind. Now I want that pore to close over. And so it's just water in, water out, water in, water out. It's not that complicated.
Or is it like black?
Yeah, there's a lot of signals going into stomata movement in particular, because if you think about it, stomata is so critical for the plant to stay alive, right? I mean, if they don't get this right, they could die because they can't like us go up to the faucet and turn the water on. And so they have a lot of controls on stomata movement. Hormones are there for sure playing into this. And I won't show it in this class. I show it in Plant Phys, but they've pretty much mapped out the pathway. And it's this big, long signal cascade that goes on in order to get guard cells to open. But the end result, all of that, excuse me, all of that signal cascade leads to, okay, it's time for water to rush in if you want the pore open. And then the water will rush out, pores will be closed over. And so it's a fine-tuned mechanism. The fancy word for what I was describing on the right is called Pulvinus tissue, which I'll explain a little bit more. But it's a special region found on some plants that, again, it's that region that will either fill up with water or water leave that area in order to cause a movement. So I'll get into that a little bit more. So the other idea, okay, turgor pressure is one way for them to move parts of their body. The other, believe it or not, is just growth. Well, so like when I was explaining this idea that abscisic acid in the root area stimulates root growth, and they go searching for water. Okay, so the roots are moving through soil in search of water. So that is a movement. It's a growth movement, and it's slower than what we tend to see with turgor movements, but it's still movement. And so you have to think about growth. Remember, growth is cell division, where one cell, divides to produce two. That's certainly growth. But also remember, cells can increase in size. That's also technically growth, right? If they just get bigger in size. And again, with growth movements, sometimes you'll see an asymmetry again, where when you zoom in on one piece of the plant and look at that tissue, some of the tissue is going through growth and another part of that same tissue is not. And so you get this asymmetrical growth in regions of the plant, and that again can cause very precise movements. And so when we think about the concepts of gravitropism, phototropism, we are looking at primarily growth movement that will demonstrate those two ideas, which I'll talk about here. Okay, so turgor movements, growth movements are ways for the plant. And so Charles Darwin and his son actually did this experiment where they were looking at ivy plants, things like that, that tend to be vining plants. And now we have videography that can show us quite clearly how the stem tip of, in particular, vining plants, they tend to kind of circle as they're growing. And essentially they're hunting for a support that they can latch onto and twist around. And so he just took some paper that had some carbon dust on it, essentially, and just let it sit there for a while, and he could trace then the movement of the shoot tip. Now we tend to do that with videography. And the fancy, excuse me, the fancy word for that is nuttation, which is that spiral. But if you think about it, it's a good action for the, especially vining plants, to take, because they're moving around in space. They're looking for a nearby support of some sort. You know, in the garden you provide a stake, but maybe they are looking for another, you know, plant nearby to latch onto. If you in the grow rack where you put your seeds that are germinating, sometimes we have a lot of bean plants, pea plants there, and they get so tangled up because we put them too close together, and then they do this while we're, you know, busy doing other stuff, and then they get tangled up with each other. So it can be quite dramatic. Okay, so these movements can be very useful, like with the vining plants. Here is an example of seeds that freshly germinated. You're looking at these water beads, kind of, so that it's instead of soil, so it's just easier to see what's happening. So they put water beads in, you know, those the gel water that you can put in to soil if you want. So, and then they put some seeds in there, and then I have video. So I believe all these video links, by the way. So as you're studying, if you put these links into your browser, it'll take you to these video clips, and they're short video clips. But they show very nicely what we're talking about here, where you can get these so-called nodding movements. And so the seed germinates, your baby, you know, the embryo starts to, you know, grow and grow, and then the seedling starts to form. Well, if you think about it, the shoot has to come out of the soil. Well, you know, shoot tissue is not terribly, it's not super strong. It's certainly not like steel. So it's got to find a way to get through the soil. If the soil, especially, is compacted, you can imagine that's going to be tough to break through the soil. And so the shoot's job is to break through the soil, get to sunlight. And that's pretty imperative, you know, like do it now. And so you can see in this video quite nicely how the stem tissue, it oscillates back and forth, back and forth. And if you think about it, if you're a plant under soil and you're trying to break through compacted soil, that kind of makes sense to kind of move back and forth as you grow up, because it loosens the soil enough where you can break through and get above ground. And so the nodding movements are actually, when you think about it, makes a whole lot of sense for these young seedlings to do this. And so that's another type of movement. You're going to see some words like phototropism, gravitropism. Tropism, that the meaning of that is like a movement. They say permanent. I guess you can get it to switch around, but permanent movements result from an external stimuli. And so phototropism, it's a growth movement in response to light. So phototropism. And you know, when you do this experimentally, you can show that the plant is either moving toward a stimulus, like toward light, for example, but it might move away from a stimulus. So a shoot and leaves will move away from gravity, right, in opposition to gravity. So it just depends on what the stimulus is. So tropism is when you start thinking about growth movements. So phototropism is this idea that we've already talked about, where the light coming from one direction, like through your window. If you have plants in the windowsill, the shoot and leaves will start to bend toward the light. And the reason they're doing that is they're trying to get their leaf blades to be 90 degrees to the incoming light. And so if, here we have a lovely bank of windows. So as the sun is coming through here, if we had plants along the windowsill here, the leaf blades would turn to be 90 degrees to the incoming sunlight. And the reason they're doing that is if you're 90 degrees to the incoming sunlight, that means your leaf blade is getting nicely saturated with light. And that's what you want for photosynthesis. So that's why they're turning to the light like that. They want to present their leaves 90 degrees to the incoming light. And it's really the auxin hormone that's doing that. So Charles Darwin and his son figured out that something's going on at the shoot tip, and it's coursing down the stem, and it's causing this phototropism. And what they finally figured out, Charles didn't do this, it came later, but what they finally figured out was that auxin is moving to the dark side of the stem. And I'm sorry, but anytime I hear the dark side, I think about Darth Vader. So many times, my son. So moving to the dark side. So what you see here on this left top picture, they've tagged auxin with this blue dye. And then what they're doing is they're hitting it with light coming from the right side. And so what you're seeing is the auxin is shifting over to the dark side of the stem. And it's causing the dark side of the stem to grow. So think about it. If you have that stem tissue with dark side is growing through cell division, that's going to cause a bending action. If the dark side grows faster than the lit side, you're going to get that asymmetrical growth pattern that will allow for the bending, in this case, toward the light. And so it's the auxin hormone that's stimulating those cells on the dark side. Now, it's not permanent in the sense that, like, if you take your plant and it's bending toward the light, you can, turn your plant, get it facing the other way, and it will, bend toward the light again. So it's not like necessarily permanent. You can manipulate that in that regard. One last idea with gravitropism, too. You have this gravity acting on the plant, and the roots want to grow toward gravity because it's telling them that's where all the soil is. You want to get into the soil and find all that lovely water. So the roots are on the left here. And what they have found out is that there are special annual plants which store starch in the roots. All these dark little balls are annual plants in the root cell. And if the root is drawing straight down, the annual plants tend to kind of settle to the bottom of the root cell, right? And so that action is what they think is causing the root to keep growing down. That's the way to go for gravity, all right? And so they give it a special term, but basically they're just anal plants. And it's the way they orient in the cell that's pressing on the cell membrane like that tells the plant, okay, keep going that way, keep growing down. And then the plant on the right there, they're just showing negative gravitropism, where the plant's like, I want to grow away from gravity. So again, growth movements away in this case. Okay, we'll finish up that idea on Friday. So please, let's hope that we can do the iClicker on Friday. So come prepared to do that. Hopefully it will work.